Hello everyone. Let's start our lecture of histology, and the topic of today is the nervous system. Nervous system. Today we will discuss the following questions. Overview of the nervous system and its functions. Then we will talk about the anatomical and functional divisions of the nervous system. And then I will give overview of some parts of the peripheral nervous system, peripheral nerves and ganglia, and central nervous system, spinal cord, and some brain structures. The nervous system is mainly composed of nervous tissue. It's a network of neurons and their supporting cells, glia. Neurons are able to generate, modulate, and transmit information between all the different parts of human body in the form of electrical impulses. The functions of the nervous system are, first of all, monitoring changes. It uses its millions of sensory receptors to monitor changes occurring in the inside and outside of the body. These changes are called stimuli, and the gathered information is called sensory input. The second function, interpretation of sensory input. It processes and interprets the sensory input and decides what should be done at each moment. And this process called integration. Next, effects, responses. It provides a response to some stimulus by activating muscles or glands, called effectors, via motor output. Next, mental activity. The brain is the center of mental activity, including consciousness, thinking, and memory. And the final one, homeostasis. This function depends on the ability of the nervous system to detect, interpret, and respond to changes in the internal and external conditions. It can help stimulate or inhibit the activities of other systems to help maintaining a constant internal environment. Anatomically, the nervous system is divided into the central nervous system and the peripheral nervous system. The central nervous system is comprising the brain and the spinal cord, and peripheral nervous system comprising the cranial and spinal and peripheral nerves, which run between the central nervous system and other tissues, together with ganglia, a collection of nerve cell bodies outside the central nervous system, and specialized nerve endings. It can be motor or sensory endings. And functionally, the nervous system is divided into the somatic nervous system and autonomic nervous system. The somatic nervous system, SNS, consists of somatic part of the central nervous system and peripheral nervous system. The somatic nervous system controls functions that are under our consciousness, under voluntary control, with the exception of reflex arcs. It provides sensory and motor innervation of all parts of the body except viscera, smooth and cardiac muscle and glands. So they are innervated by autonomic nervous system. The autonomic nervous system consists of autonomic part of the central nervous system and peripheral nervous system, and the autonomic nervous system provides efferent involuntary motor innervation to smooth muscles and the conducting system of the heart, glands. It also provides afferent sensory information from the viscera. It can be pain or autonomic reflexes. And the autonomic nervous system is subdivided into three parts: sympathetic division, parasympathetic division, and enteric division. The presynaptic neurons of the sympathetic division are located in the thoracic and the upper part of the lumbar portion of the spinal cord. The presynaptic neurons send axons to the vertebral and paravertebral ganglia. The paravertebral ganglia in the sympathetic trunk contains the cell bodies of the postsynaptic effector neurons of the sympathetic division. The presynaptic neurons of the parasympathetic division are located in the brainstem, here, and sacral spinal cord, here. The presynaptic parasympathetic neurons send axons to visceral ganglia. And the ganglia are located in or near the wall of abdominal and pelvic organs. So, nearby these organs, or in the walls of these organs. And the visceral motor ganglia of cranial nerves, here, of the parasympathetic division, they also contain the cell bodies. The sympathetic and parasympathetic divisions of the autonomic nervous system often supply the same organs, like you can see here, from this part, and from this side and this side. In these cases, the actions of the two of these divisions is usually, but not always, antagonistic. For example, sympathetic stimulation increases the rate of the cardiac muscle contractions, and the parasympathetic stimulation reduces this rate. The enteric division of the autonomous nervous system consists of the ganglia and their processes that innervate the alimentary canal. It controls motility, contractions of the gut wall, exocrine and endocrine secretions, and blood flow through the gastrointestinal tract. It also regulates the immunologic and inflammatory processes. The enteric nervous system can function independently from the central nervous system, and it can be regarded as the brain of the gut. However, digestion requires communication between the enteric neurons and the central nervous system anyway, and it is provided by the parasympathetic and sympathetic nerve fibers. Ganglia and postsympathetic neurons of the enteric division are located in the lamina propria, muscularis mucosa, submucosa, muscularis externa, and the subserosa of the alimentary canal, from the esophagus up to the anus. So first we begin with the considering of the structures of the peripheral nervous system. Peripheral nerve. The bulk of the peripheral nerve consists of the fibers and their supporting Schwann cells. So here you can see examples of myelinated fibers and unmyelinated fibers. The individual nerve fibers and their associated Schwann cells are covered by connective tissue, organized into three distinctive components. Each have its special morphologic and functional characteristics. The one that covers each nerve fiber includes loose connective tissue surrounding these fibers. Each fiber is called endoneurium. The perineurium, here you can see an example, is a specialized connective tissue that surrounds each nerve fascicle, so the groups of the nerve fiber. Why it is a special tissue? The perineurium may be one or more cell layer thick depending on the nerve diameter. The cells that compose this layer, called perineurial cells, they are squamous. Each layer exhibits a basal lamina on both surfaces, and perineurial cells possess receptors, transporters, and enzymes that provide for the active transport of substances across these cells, across themselves. The cells also contain actin filaments that makes them contractile. Besides these perineurial cells, only fibroblasts and small number of resident macrophages, as well as occasional mast cells, can be found in this perineurial component. The third layer is the epineurium, this one. It covers the whole nerve, and it includes the dense irregular connective tissue. Adipose tissue is often associated with epineurium, especially in the large nerves. Peripheral nerve fibers, so these fibers, they are classified based on their diameter, signal conduction velocity, and the myelination state of the axons. So this classification applied to both sensory and motor neurons, and the fibers of the A group also classified into 4 subgroups. A alpha fibers, they are primary receptors of the muscle spindle and Golgi tendon organ. A beta fibers, so it's the somatic motor and proprioception. A beta fibers act as secondary receptors of the muscle spindle and contribute to cutaneous mechanoreceptors. So it's touch and pressure. A delta fibers are free nerve endings that conduct painful stimuli related to pressure and temperature. And A gamma fibers are typically motor neurons that control the intrinsic activation of the muscle spindle. Fibers of group B, they are myelinated with small diameter compared to the A fibers, which have larger diameter. And they have lower conduction velocity. The primary role of B fibers is to transmit autonomic information. And the fibers of C group, they are unmyelinated. They have smaller diameter and the very low conduction velocity. The lack of myelination in the C group is primary cause of their such a slow conduction. The spinal nerve is an example of a peripheral nerve. It has afferent sensory part. These fibers enter the spinal cord via the dorsal roots, while afferent motor fibers, they leave the spinal cord via the ventral root, here. Dorsal and ventral roots merge to form the spinal nerves. So here the spinal nerve that contains both somatic motor neurons, somatic sensory neurons. So also it contains some autonomic unmyelinated fibers. As the spinal nerve travels into the periphery, they split into some branches. Vascularization of the nerves. Arteries pass into the epineurium, so here, and form arterial network beneath the perineurium. They give off the capillaries to the endoneurium. Tissue at level of endoneurium is quite poorly vascularized and also depend on the diffusion of the nutrition from the perineurium. And we observe a lot of anastomosis in the vessels of the nerve. So here are some histological illustrations of peripheral nerve. So this micrograph shows the typical appearance of a medium-sized peripheral nerve in a transverse section. So it consists of some fascicles here. Each fascicle contains many nerve fibers. They are covered by perineurium, and the whole nerve is enclosed into the epineurium. Blood vessels of various sizes you can see here, here, here. Next is an electronic micrograph of the fascicle with fibers of different sizes. Here you can see axons and myelin cover and the different sizes of the fibers. Some fibers of the loose connective tissue can be seen, the endoneurium, and the perineurium covers all this fascicle. And here is the osmium stained slice, the transverse section, and it also contains many fibers, myelinated fibers of different sizes, the endoneurium, and it also contains some unmyelinated fibers, small ones. You can see the perineurium that covers the whole fascicle, and adipose tissue cells, the adipocyte, in the connective tissue outside the perineurium. So, ganglia are aggregations of nerve cells outside the central nervous system. The ganglia can be broadly categorized into two groups that are sensory and autonomic ganglia. The sensory ganglia are relating to the somatic neurons, somatic nervous system. They include dorsal root ganglia and cranial nerve ganglia. So the dorsal root ganglia and the cranial nerve ganglia. And there are also autonomic ganglia. The dorsal root ganglia or spinal ganglia, it's the place where the cell bodies of the sensory or afferent nerves are located. And the cranial nerve ganglia contains the neurons of the selected cranial nerves. Autonomic ganglia contain the cell bodies of the autonomic nervous system. Here. Neurons in the cranial nerve and dorsal root ganglia are pseudo-unipolar. You can see here example of dorsal root ganglia, and the neurons are pseudo-unipolar. So the cell body are usually round in shape, the centrally located nuclei. There is a wide range of sizes of these neuron cell bodies, with the largest having axons that are heavily myelinated and lightly unmyelinated or lightly myelinated or unmyelinated smaller size neurons, and that carry, for example, pain and temperature information. So here is example of the perikaryon, the cell body of one of the neurons in the ganglion, and it is surrounded by numerous satellite cells, the glial cells. They regulate the extracellular ionic environment. And the Schwann cells, as I told you previously, they provide the myelin for the myelinated axons. The cranial nerve and dorsal root ganglia are surrounded by connective tissue capsule that continues with the dorsal root, epi, and perineurium. Autonomic sympathetic ganglia. Here is an example of histological slide, and this sympathetic is this part. The ganglia are located here. So they found in the adjacent to the vertebral column. They have a similar structure to that of somatic sensory ganglia, with some differences. The ganglia neurons are multipolar compared to pseudo-unipolar neurons in the dorsal root ganglia. It means that their processes, they are more widely spread, and it's less satellite cells around each cell body due to numerous of processes around it. As seen as this micrograph, the nuclei tend to be eccentrically located, and the peripheral cytoplasm contains a variable quantity of brown stayed granules, so lipofuscin granules. They represent cellular debris and residual bodies. The satellite cells are smaller and irregularly placed. In autonomic parasympathetic nervous system, the cell bodies of the terminal effector neurons are located near innervated organs, and some ganglia which are embedded within the walls of the organs, which are called intramural ganglia. Commonly, only a few neuron cell bodies are clumped together with supporting cells, and they form very small ganglia. And they are scattered in the supporting tissue. So here on the micrograph, you can see small ganglion from the wall of the gastrointestinal tract. The ganglion cells are recognized with their large nuclei, dispersed chromatin, and prominent nucleoli. Here, for example, here. In other ganglia, the neurons are surrounded by, like the same as in the other ganglia. They're surrounded by the glial cells and also fibers, afferent and efferent nerve fibers. So, let's go to the central nervous system. So the central nervous system contains of the brain, located in the cranial cavity, and the spinal cord, located in the vertebral canal. So the central nervous system is therefore protected by the skull and vertebrae, and it is surrounded by 3 connective tissue membranes called meninges. The surface of the nervous tissue is covered by a delicate layer called the pia mater, containing collagen fibers and fine elastin fibers. Also, some occasional fibroblasts can be found in this layer. This layer is separated by a basement membrane from the underlying parenchyma of the central nervous system. So here is this pia mater. The arachnoid matter is a thicker fibrous layer, this one. And here in the spinal cord. The pia and arachnoid matter are continuous, and they have the same oranges of the single layer of mesenchyme that surrounded the developing brain. So they may be considered as one unit, the pia arachnoid unit. The space between the pia and arachnoid is called subarachnoid space, this one. Arteries and veins can pass from the central nervous system to this arachnoid space, and they are loosely attached to the pia mater. So external to the arachnoid matter is a dense fibroelastic layer called the dura mater. In the cranium, the dura mater merges with the periosteum, here, merges with the periosteum. And around the spinal cord, the dural matter is suspended from the periosteum of the spinal canal by denucleated ligaments. Here, this one. So, I want you to remind the central neuroglia, which play a very important role in the support of the neurons of the central nervous system. So the glial cells aid in transferring nutrients from the capillaries to neurons. They maintain the blood-brain barrier, regulate the intercellular environment, provide myelin insulation for axons, and provide mechanical support to neurons. Also, they act as phagocytes to remove pathogens and dead neurons, and play a role in presenting antigens in the immune system. They can perform some other functions as well. It is thought that there are as many as 10 times the number of glial cells as neurons in the nervous system. So they are very numerous. And the major types of the central nervous system glial cells are astrocytes, oligodendrocytes, which produce myelin, microglia, which act as the phagocytotic cells and elements of immune system, and ependymal cells that line the ventricles of the brain and the central canal of the spinal cord. We should also mention the blood-brain barrier. It protects the central nervous system from fluctuating levels of electrolytes, hormones, and tissue metabolites that circulate in the blood. The blood-brain barrier develops early in the embryo development through the interaction between glial astrocytes and capillary of endothelial cells. So the barrier is created largely by the elaborate tight junctions between the endothelial cells, so endothelial cells with tight junctions. The barrier is provided by the capillaries, which are continuous. Other components of the barrier are basement membrane of these endothelial cells and the astrocyte processes that called also end feet, and they form the contact with the capillaries. Okay, let's consider the spinal cord. The structure of the spinal cord is quite similar throughout its length, but it has some special form at different layers. It divided in the 31 segments, 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 1 coccygeal. And each segment is connected to a pair of spinal nerves. So, segment, pair of spinal nerves. Here you can see 4 regions: cervical, thoracic, lumbar, and sacral. In transverse sections, the central mass of gray matter has a shape of butterfly. The ventral horns are quite prominent and contain the cell bodies of the large motor neurons. You can see here example. The gray matter here. The dorsal horns contain the cell bodies of small second-order sensory neurons. These bodies relay upward sensory information on pain and temperature, and also they participate in the spinal reflexes. Small lateral horns are found in the thoracic and upper lumbar levels. So here the lateral horns. The cell bodies of sympathetic nervous system, efferent neurons, are located there. So the groups of nerve cell bodies in the gray matter are called nuclei. Nuclei of the central nervous system are morphologic and functional equivalents to the ganglia of the peripheral nervous system. And synapsis occurs only in the gray matter. The white matter consists of the ascending tracts of sensory fibers, ascending tracts, and descending motor tracts, passing up the spinal cord towards the brain, and more fibers enter and leave the cord, so the volume of white matter increases progressively from the sacral to the cervical region. So you can see very little white matter here, and then the fibers enter the white matter, and finally it's really big volume here. So the brain and the spinal cord, they have also cerebral spinal fluid that occupies the space between the meningeal layers. The structure of the brain. The brain is thoroughly subdivided in the cerebrum, cerebellum and the brainstem. In the brain, gray matter forms our outer covering layer, or cortex. And the white matter forms inner layer or medulla. The cerebral cortex that forms the outermost layer of the brain contains nerve cell bodies, axons, dendrites, and central glial cells, and it's a site of synapses. In a freshly dissected brains, the cerebral cortex has a gray color, so that it's named gray. In addition, the islands of gray matter, called nuclei, can be found in the deep portions of the cerebrum and cerebellum. The white matter contains only axons of the nerve cells and plus associated glial cells and blood vessels. So the axons in fresh preparations appear white. That's why it's called white matter. So let's consider in detail the structures of the cortex, of the cerebellum and the cerebrum. First, the cerebellum. The cerebellum is a large complex structure that lies beneath the posterior portion of the cerebral hemispheres. It coordinates muscular activity, maintains posture and equilibrium. The cerebellum, it has cortex that forms a series of deeply convoluted folds. Here you can see these folds. They are also called folia. There are 3 layers in the cortex of the cerebellum. The outermost molecular layer, the Purkinje cell layer, and the granule cell layer. The molecular layer contains primarily axons and dendrites, with only very few cell bodies of 2 main types of neurons. These are stellate cells and basket cells. The ganglionic layer or the Purkinje cell layer, it's a layer between the molecular and granular layer. And the Purkinje cells have very large cell bodies, relatively fine axon that entering down through the granular layer. So here you can see example. Axon goes down to the granular layer. And it has a very extensively branching system of dendrites. The axons of the Purkinje cells provide the only efferent pathway to the deep cerebral nuclei, and thus Purkinje cells constitute the sole output of the motor coordination in the cerebral cortex. The granular layer contains an enormous number of very small, tightly packed granule cells, and they are dendrites. So you can see example of granule cells, dendrites, they are all here. The axons of granule cells, they rise vertically and then form the T-split, T-junctions, that branch and run parallel to the surface. and they synapse with dendrites of the Purkinje cells. Besides the granule cells, also you can find in this layer the Golgi cells and brush cells. So here we can again look at the molecular layer. There are stellate cells, basket cells here, and mostly the processes of the neurons. Then the ganglionic layer or Purkinje cell layer with Purkinje cells, and the granular layer with numerous granular cells, and also some Golgi cells here. So the cerebellar cortex contains fibers that are termination of afferents, the cerebellar afferents. The afferent fibers, they enter the cerebellum from the brain stem, and then they pass there the central white matter and make complex connections with the cells in the cerebellar cortex. The histologically, there are two distinct types of fibers. The Mosey fibers, here you can see the Mosey fiber, they take place within structures called glomeruli. Here is one glomerulus. And the terminals from the Golgi cells also form the structures. Here the Golgi cell. So the granule cells, Mosey fiber and Golgi cell, they form the glomerulus. Afferent impulses through the Mosey fibers, they pass from granular cells, so through granular cells, and they can reach several Purkinje cells. Another type of fibers, climbing fibers, they pass through the granular layer and the Purkinje cell layer and reach the molecular layer, where they climb on the dendrites of the Purkinje cells, and they wind around them, establish numerous synapses with them. Both Mosey fibers and climbing fibers, they are excitatory. And then we come to cerebrum. The cerebral cortex is the outermost layer of the brain, and it is associated with the highest mental capabilities. The cerebral hemispheres, right and left, they consist of the convoluted cortex of gray matter, the thickness about 3 mm, and overlying the central medullary mass of white matter. Here. The surface area of the cortex is increased by the convolutions, which are separated by fissures. The cerebral cortex consists of neurons, nerve fibers, and neuroglia. Histologically, the neurons of the cerebral cortex can be divided into 5 main groups. The pyramidal cells, these ones, they have pyramid-shaped cell bodies with the apex directed towards the cortical surface. And from the apex, a thick branching dendrite passes towards the surface, where it arrays of fine dendritic branches. An axon arises from the base of the cell, here, and passes into the white matter through the case. So in the case of the small superficial cells, the axon may synapse in the deeper layer of the cortex, and the short dendrites arise from the edges of the base and ramify laterally. The size of pyramidal cells varies, with the smallest sit superficially and the largest motor neurons of the motor cortex, also called bed cells. They are the largest pyramidal cells in the cortex. Then the stellate or granule cells. Stellate cells. They are small neurons with a short vertical axon and several short branching dendrites. They have a shaped body of star. The routine histological methods, this cell look like small granules. That's why they are named like granule cells. Next type is cell of Martinotti. Here. It's a small polygonal cells with few short dendrites. The axon extends towards the surface and bifurcates to run horizontally, usually to the most superficial layer. Bifurcation. Next, the fusiform cells. Here. These are spindle-shaped cells oriented at right angles to the surface of the cerebral cortex. The axon arises from the side of the cell body and passes superficially. Here. Dendrites extend from each side of the body and branch into deeper and more superficial layers. And the horizontal cells, which are small and spindle-shaped but oriented parallel to the surface. They are the least common cell type and are only found in the most superficial layer. Their axons pass laterally and synapse with the dendrites of the pyramidal cells. So the neocortex is arranged into the 6 layers with different neuron morphology. And here you can see the diagram of all these layers with different staining, the Golgi staining, Nissl staining, and Viger staining. So this is to stain the fibers. There is a plexiform or molecular layer, the most superficial. It's also called lamina molecularis. It contains the dendrites and axons of cortical neurons, making synapses with one another, and nuclei are sparse and of neuroglia and horizontal cells of Cajal, or the horizontal cells, which we considered previously. Outer granular layer, or lamina granularis externa, or the external granular layer, it's also called. It contains dense population of small pyramidal cells. Small pyramidal cells. Also, there are various axons and dendritic connection from the deeper layers. Next, the external pyramidal cell layer, or lamina pyramidalis externa. The pyramidal cells are of moderate size and predominant in this broad layer. The cells increasing in size deeper in the layer And Martinotti cells are also present in this layer. Inner granular layer. Lamina granularis interna. This layer consists mainly of densely packed stellate cells. Next, internal pyramid layer or ganglionic layer, lamina pyramidalis interna, large pyramidal cells and small number of stellate cells as well as cells of Martinotti can be found in this layer. And the huge pyramidal cells, the bed cells, they are very prominent here. Next, the multiform layer. The multiform layer, or lamina multiformis, is named for the wide variety of different morphological forms of the cells, of the neurons. It contains numerous small pyramidal cells and cells of Martinotti, as well as stellate cells, especially in the superficial part of this layer, and fusiform cells in the deeper part of this layer. And in many layers, the fibers go in such bundles in parallel, directed parallel, and vertically, so connecting different parts of the cortex and cortex with underlying structures. So, and finally, I will remind you the development of the nervous system. So the nervous tissue forms from the neuroectodermal plate on the dorsal side of the embryo, dorsal side here. The neural plate forms three parts. It is the neural tube, the ganglion plates or neural crest, and also the neural placodes. The neural plate thickens, begin to invaginate here, and then it invaginate in the underlying mesenchyme, forming the neural folds and neural groove. The process of neurulation continues with the closing the ridges, the neural folds, and forming neural tube. As the neural folds elevate and fuse, the cells of the lateral border, they migrate outside and give rise to the neural crest. The neural tube gives rise to the brain and spinal cord. The cells of ganglia, peripheral nerve system, and part of the peripheral nerves, pigment cells of the skin, the cells of the Apud system, and the adrenal medulla, they develop from the neural crest. In the cranial section of the embryo, on the sides of the neural tube, neural placodes are formed. They give rise to the nuclei of the 5th, 7th, 9th, and 10th cranial nerves. The central hollow space of the neural tube later forms the fluid-filled brain ventricles and the central canal of the spinal cord. So development of the brain. The cephalic end of the neural tube forms 3 dilations here. They called the primary brain vesicles. They are prosencephalon or forebrain, mesencephalon or midbrain, and rhombencephalon or hindbrain. During the subsequent divisions, the secondary brain vesicles they transform further. Like the procephalon gives the secondary vesicles telencephalon and diencephalon. The telencephalon then is developing into the cerebrum, and diencephalon is forming the eye cap, thalamus, hypothalamus, and epithalamus. The mesencephalon give rise to the midbrain. Rhombencephalon or hindbrain, it then forms the metencephalon, which give rise to the pons and cerebellum, and myelencephalon, which gives rise to medulla oblongata. So, this was the overview of the nervous system. Thank you for your attention and have a nice day.